morphogenesis of embryos in early development. And so, this is a very, very general introduction. I apologize for that. We're interested to see how essentially you start with a fertilized oocyte or a fertilized egg, and what you're ending up kind of with another organism. And this is very largely spoken what we try to understand. Now, there are two processes that, by which the oocyte develops into an adult organism. One is patterning, cell phase specification in space and time, and the other one, this is the process we are particularly interested in, is morphogenesis, how the organism takes a specific shape, which is characteristic for a specific species. Now, to give you a here, this is the first deviation from my normal talk, and to give you a little bit of an introduction about my CV and scientific pedigree. It's not particularly exciting, it's pretty standard. I studied biology in Munich in Germany, and from there on and did my PhD in the lab of Christiane Lustan-Vollert at the MPI in Tübingen. And from there, after the PhD, did a postdoc, like many of you are doing or have been doing. And I did that at the University College in London, in the lab of Steve Percy. And from the postdoc, I applied and became a PI, principal investigator, and group leader at the MPI Molecular Cell Genetics in Dresden. There I stayed nearly 10 years, but luckily we have been at Max Planck, we had the possibility to stay for 5 years plus 4 years, so 9 years in total, and if you're still not away, then you give you another year, you can stay for 10 years. And after that, I became a tenured professor at the Institute of Science and Technology in Austria, which is close to Vienna in Austria. And that's it. There's not much more about it. What I do is not only science, and I put these pictures in here just to illustrate a few things. You shouldn't take the title too serious. I'm interested in running occasionally, and playing cello, as already pointed out, and biking on the sometimes different types of bikes as well. When I joined the Institute of Science and Technology in Austria, I faced essentially this situation here. It was a psychiatric hospital before, and they pulled it on, and they wanted to build a new institute. There was essentially nothing other than rubble and an empty building. So the risk of joining this place was quite considerable, because there was no infrastructure. But the gain was sort of scaling the risk in case this is going to work out, you have really achieved something. And luckily we got into a big institute at the end, which is now over 100 groups and more than 1,200 scientists working there. It's a very interdisciplinary research environment. We have biology, we have physics, we have chemistry, material science, evolutionary biology, name it. It's pure mathematics. It's all there on campus. And it's for European research institutes very unusual that everything is on our place. So it's a campus university. Okay, and then, nothing I do would be possible without that I'm doing it. And this is, at the point when we started to be at the Institute of Science and Technology in Austria. And then we had a period of, Corona where things went hard for experimentalists, particularly. And now we're back in the lab again. And now I tell you a little bit about science, if that's okay. Just to give you sort of an overview of what we study and how take or the lab has taken a specific research question. So as I said, we are interested in seeing how a fertilized oocyte transforms into an embryo with a clear anterior posterior polarity, left right polarity, and the closed ventral polarity. And how do you form essentially out of a seemingly unstructured cluster of cells something which looks like a proper embryo? That's really what we are trying to do. And we were looking from a perspective at the interface between mechanics, physics, and biology. And we were interested to see how mechanical forces are being generated which underlie morphogenesis in the embryo. So we looked at force generation mostly in the framework of actin, myosin, and actin-myosin flows, generating forces. Put here one example, very early process in development when the fertilized egg is segregating the ovoplasm from the yolk run. The yolk run is blue stuff and the ovoplasm is the yellow red stuff. And there's a complete segregation of these two phases. And this is driven, like many other processes in early development, by flows of actin arising. These flows are directed from the ventral flow up to the animal flow. They take along the ovoplasm and they leave behind the yolk run. And this leads eventually to a complete segregation of ovoplasm in the very early development. We also got interested in how mechanical forces are transmitted and how they are balanced within the embryo. And one typical case where mechanical force transmission becomes important is due to contact formation and cells forming in contact. And what we sort of worked out, and this goes back on nearly 40 years, is the function of cell adhesion molecules versus, again, the acromycin cytoskeleton. And what we showed is that the most important thing to form a large contact is to disassemble the acromycin at the forming contact, leave it intact on the outside. And then the balance of these tensions, which are exerted by the acromycin cytoskeleton, determines how big this contact angle becomes and how large the contact is in the contact angle. And talking about forces, and you look at living matter, then one process very specific for living matter is mechanicization. That cells can not only exert mechanical forces, but they can receive mechanical forces and respond to them. So we got a number of years ago into the process of mechanicization. I put in one example where you're looking at an epithelial cell, which is sitting here, which is about to divide. And what we're doing is we're asking if forces and the anisotropy or the anisotropy of mechanical forces in the plane of the epithelium has an influence on the direction the cell is dividing. And what you see here in white is the mitotic spindle. And what we're doing is experimentally we're inducing the ectopic axis tension within the plane of the tissue, and we find that the mitotic spindle can read this new axis of tension, this ectopic axis of tension, which directs the division of the cells. Okay, so this is just sort of a very rough overview about what we're interested in, what we're working on. They give you now one example which has sort of kept us busy during the entire period of our lab, which started in 2001. And so what we wanted to understand when I started my group at CBG in Weston is we wanted to understand how this tissue starts to spread, the plasmid starts to spread over the yolk cell. And this process is called equity or doming. And it appears as if the yolk moves into the tissue, but essentially this tissue moves and spreads around the yolk cell. And what we wanted to know what are the forces generating, what is the mechanical underpinning of this process. And essentially the first thing we noted is during this process there's a very large acromycin cable forming in the rim of this tissue right here. And this acromycin cable, once it has moved over the equator of the yolk cell, constricts, and by constricting it pulls on the Shiv cells, which is this Shiv here cells, and pulls it over the yolk cell. And that's sort of a very intuitive process how you can trigger spreading of tissues. And it's a process actually which is quite similar to what you typically find in medically relevant processes like wound healing. This is a case in Drosophila where you see that there's an acromycin cable forming this axis of herstrin and pull these cells together. It's not only that, but this is certainly a contributing factor. And another process in development for all the Drosophilus is also closure, where you have again a contractile cable which acts as a herstrin and leads to closure of the dorsal opening. Now what we wanted to know first is what are the force-generating processes which lead to the spreading of the plasmid of the yolk cell. And what we did is we sort of constructed a very, very simple mechanical model of this process, and we assumed that there are two main forces. One force is generated by this acromycin contractile band. Then there might be another force which is generated by regulation of surface tension, the spreading of cells which might modulate the surface tension, which makes it easier for tissue to spread. And there might be active integration of cells which leads to a thinning of the tissue and spread of the tissue. these were sort of the, very globally spoken, the main cause we thought might be in these tissues. Now, just to introduce the main tissues, when you look at it, we have deep cells, we have surface cells, which are called the enveloping layer cells. This is simple spring, the clear cells, and they are connected to the York cell. The York cell is this big clump of York granules, and they make tight junctions with the York cell. And the York cell contains a very thin layer of cytoplasm which contains all known cytoskeletal elements, intermediate elements, actin, myosin, microbes, you name it. And what we noted, and other people noted before us already, is that there is a contractile cable of acromycin forming at the point where the EDL makes contact with the Y cell, the York incision layer, this thin cytoplasmic layer. And what we speculated, again, as many other labs have done that before, that this might be one of the key force-generating processes which lead to spread of the EDL and lose the entire tissue over the outside. Now, this is the process I showed you before already. And what we did in this part of the work, we analyzed how this acromycin ring or bell can actually act on exerting forces. We came up with two motor processes which it can do. One, it can act as a cable constriction motor that's essentially just constricting around its circumference, and by constricting around its circumference, once it has moved over the equator, it just needs to constrict and it will pull it out. I think that's pretty clear. But we also realized that this cannot be the only process, that there's a flow friction motor as well that acromycin within this valve here is not only constricting around its circumference, but it is also constricting along its length, or its width in this case here, along this axis. And by constricting along this axis, it induces, quite often in acromycin networks, it induces flows of actin. And these flows of actin are directed from this end of the actin network, which is uncoupled, to the other end of the actin network, which is coupled to a junctional complex here. So you get a retrograde flow of actin in this orientation here, which, if you flow and you resist by some friction to anything which is adjacent to it, York values, you name it, will exert a pulling force in the opposite direction and produce the tissue elements. So we have a cable constriction over and a flow friction. So that sort of was setting the main force generating process, and then we asked, what would happen here with the surface cells and the deep cells? Is it essentially radial intercalation of cells which thins the tissue in one direction and extends it in the other direction? Or does it have maybe to do something with these surface cells which might be actively spreading, and they were reducing surface tension and actively contributing to the spreading of the tissue? What we did then is we in this case, this is Marita, a postdoc in the lab, what he did, he looked at these deep cells, and he did one sort of key experiment, and often it is one experiment which sort of gives you a clue into how this process works. He thought the way he can distinguish between surface cells and deep cells is essentially just getting rid of the deep cells and asking if the process will still work in the absence of deep cells. So the absence of this cluster of active interaction. He did an experiment which not many people can do, is he took a pipette, he took out these deep cells to a large degree, he depleted the deep cells, and then he was asking if this depleted cluster can still undergo spread. And next to the experiment, we have a few cells left in here, and to our big surprise initially, this depleted tissue still undergoes spreading for the York cell, indicating that active contraction of these deep cells is not really required for the spread of the tissue. So we were left with the surface cells, and Hikoshi was left with the surface cells, and he was asking, would active spreading of these surface cells be critical for the spread of the tissue? And again, he did then an experiment which is a very difficult experiment. And I can jump over that here quickly because for the sake of time, he did a tissue transplantation experiment where he took cells from a wild-type embryo, for example, and puts it onto a wild-type host embryo, and he can take tissue from an embryo where the spreading of the surface cells is genetically inhibited, so these cells cannot spread. And then he was asking what would happen for tissue spreading in case where you had EBR cells which can spread versus EBR cells which cannot spread. And this is an example where he took mutant tissue, into a wild-type embryo. In this case, he takes out wild-type tissue into a mutant embryo. And what you see is that once you have cells which cannot spread, which cannot undergo a kind of spread in surface cells, then this spreading process of the tissue is inhibited, and you can induce ectopic spreading in cases where you normally not spread, and then you get this local tissue spread. Was this bell for me already? Okay, I should speed up a little bit. How many minutes do I have? 10 minutes. 10 minutes, okay. So what the logical conclusion from that is that the spreading application of deep cells is not important. What you need to really look at is the spreading of these surface cells. And so what we then saw is perhaps this contraction of the deep cells is not really required as force generating process, but still it is a material which needs to be deformed. And using the material properties of this tissue is quite important to be permissive for spreading the tissue. And this was an assumption which Nicoletta Petrigo's lab took on. She asked if this is part here, would changes in the material property of the deep cells be important for the spreading of the tissue? And what she noted then, I can go very quickly through that, is that the tissue in the center of the plastidome undergoes a fluidization right at the point when the tissue begins to spread. And that's sort of an indication that this tissue needs to become more fluid in order to properly spread over the yards. And this is viscosity measurement which he did. And then she did experiments to see if that is indeed the case, that you need this fluidization of the tissue. And again, by heterotypic deep cell transplantation experiments, very similar to what Hikashi did on the surface. Did now deepen the tissue, and she could show that when you put deep cells into the middle of the embryo, which cannot fluidize for various reasons, genetically or mechanically, then the spreading of the tissue is very similar to the surface cells. If surface cells cannot spread, if the tissue cannot spread, if deep cells cannot fluidize, then the tissue is not spread either. The question that she asked is, how would the tissue undergo this fluidization? And she looked at a process which is familiar to all of you, and that's cell divisions. And these cells are undergoing synchronous cell divisions, and while undergoing these synchronous cell divisions, during a division there's mitotic grounding, there's a transient loss of cell-cell contact. And what she found is that these synchronized divisions within the deep cells are critical for the fluidization of the deep cells, the fluidization of the deep cell tissue, and thereby facilitate the spread of the tissue. And again, I should be relatively quick on that. That's what she proposed. There is a transient fluidization of the deep cells, and this transient fluidization is driven by synchronous cell divisions, and these synchronous cell divisions is required for tissue spreading. The fluidization is required for tissue spreading. I might go over that, jump over that, and just come to a conclusion. What we showed in this work here, and this was the last work on the deep cells, is that the formation of these deep cells, their contact formation forms networks of contacting cells, and this network of contacting cells breaks down if a specific connectivity within this network is being achieved. These are networks of connecting cells in the deep cell tissue, and then you have a transition, a phase transition from a viscous tissue into a fluid tissue once the connectivity goes below a critical value between these cells. But I wouldn't have enough time to go really into detail on that. So essentially what Nicoletta then showed together with Eduard and Nesso, that this tissue fluidization is essentially very similar to a phase transition from a very rigid tissue into a fluid tissue, depending on the connectivity of these cells, at a critical value of connectivity of these cells. Now this sort of, we thought at this point we have solved the problem. We know now it's surface cells spreading, it's to a certain degree fluidization of deep cells, and we have this acromizing ring, and everything is sort of done until another postdoc joined the lab, Krishna. She's still in the lab, and she did one sort of critical experiment where she thought, what would happen if we are just taking the entire plastidome away? So surface cells, deep cells, nothing is left. Nothing should really happen. Now this is an embryo which has no plastidome anymore, it's just a yolk cell, and this is the proper embryo. And what she found is that the depleted embryo undergoes spreading, but there's no tissue. So that's a very interesting question because it tells you that the yolk cell itself is probably triggering the spreading process, and it doesn't care if there's a plasmon sitting on top of it to spread. It has it's an inherent property of the yolk cell and the contact, this contact between the formoplasmon and so the inside and outside of the yolk cell, which really trigger this spreading process. Very interesting. Tushna is working on it now, and she has evidence, and I cannot tell you any more details about it, is that this part of the yolk cell is apical, this part of the yolk cell is basal, and what you're looking at is a process very similar to an apical constriction of an epithelial cell. And this is essentially one big epithelial cell which goes apically constricted. That's the idea Tushna is going to do. Okay, so I'm pretty much at the end. I should be at the end. And again, this is the supporting agencies and that's the plan. And thank you for your attention.